Hello, baby. Yeah, this is the Big Bopper speaking. The aim of today's experiment is to demonstrate or illustrate the process of osmosis in plant tissue. Now, I think you know what osmosis is, but I'll give you a quick definition of it. Osmosis is the movement of water molecules from an area where there's a high concentration of water molecules to an area where there's a lower concentration of water molecules through a selectively permeable membrane. And that's why osmosis is a form of diffusion. Diffusion, you'll remember, is the movement of any substance from an area of a high concentration of the substance to an area with a lower concentration. So there's not a selectively permeable membrane involved. So what I need to demonstrate the process in plant tissue is 2 potatoes, and I've peeled the potatoes, both of them, and I've cut them like a straight here, and then I dug a hole in each of these potatoes. It should be as deep as possible, but don't cut a hole in the potato's tissue. Then the solutions will just simply leak out. And yeah, I've done the same thing. So I've got 2 similar setups, but in the case of A, that is my experiment, and B, that's my control. I'll explain why that is. So what I then do is I add to each of the beakers a similar amount of distilled water. I then put the potatoes in, and then I add, in the case of this experiment, I prepare a 20% sucrose solution by adding 4 teaspoons, more or less 5 grams. 4 times 5 is 20 grams of sucrose to 100 milliliters of distilled water. And then I pour some of the solution here into this hole of the potato. And you'll see here I've marked the level of the sucrose solution with a pin here. Now you'll see that yes, yesterday the level of the water was lower than it is today, and I'll explain that just now, because you can see now the other end of the pin is like covered with the solution. I've added some food coloring to the solutions on the inside just to make them clearly visible to me. It doesn't affect the process of osmosis though, so that's why I can do it. In the case of the control, I also add, like I said, I've added distilled water here, but I also add distilled water here into the hole. I pour it in here, and then I also mark the level of the water with this pin. And you'll see, if you look carefully, you'll see, if I turn it like that, you'll see that the level of the water on the inside has already, it's more or less remained the same overnight. Where in this case, as I've mentioned, there was a rise in the level of the water. And can we explain that? Now, we can explain it in terms of 3 different sets of terminologies. First of all, we can talk about osmotic potential. And we'll say because osmotic potential of a solution is the ability of that solution to absorb water through a selectively permeable membrane. And you can see, because I've added the sugar solution or the sugar molecules to this solution here on the inside, it's got a very high osmotic potential, and that's why its ability to absorb water through the selectively permeable membrane that's presented by the potato is higher than that of the distilled water on the outside. In this case, my control, here you can see the osmotic potential on the outside, on the inside of the potato is similar. So that's why there was no net movement of water molecules from inside out or from the outside to the inside. That's what I called an isotonic solution. And while I'm talking about isotonic, I can explain the words isotonic, hypertonic, and hypotonic. In this case, like if I have to explain it in terms of hypotonic, I've got a solution or a liquid with a hypotonic concentration here. It's a hypotonic solution. On the inside there's a hypertonic solution, and that's why water is moved from here to the inside. Like I said, here it's isotonic. So if I move to this diagram, then I can see that here we've got an hypotonic solution. Eh? That is the selectively permeable membrane of the potato, and here we've got an hypertonic solution. So then the water moved from here to there. I can also explain it in terms of water potential. Now, water potential is the potential of a solution to, or like we can almost say, push water from here to there. So here we can see, because there's some sugar here, the solution here, the water here, it's got a very high water potential, and then the high water potential will cause water to move into this hole here, and there you'll see the level has increased overnight because there was some water added to the solution. It can be quite dangerous if we look at an example from humans and animals, where you can see here I've got 3 sets of red blood cells, and these blood cells have been placed in a hypertonic solution. And what happened then, because the solution of the concentration of water on the inside of the red cells was higher than that on the outside, so then it lost water through the selectively permeable membrane of the red blood cell. So that's what's called a hypertonic solution. The hypertonic solution is on the outside of the red blood cells. If we talk about the hypotonic solution, here you'll see water has moved into the red blood cells, and that can cause them to burst because they have a very thin membrane, and it doesn't have a cell wall that can limit the breaking up of the red blood cells. So then you can see it burst. It's like when someone, for an example, when someone gets a stroke, and then blood is then pumped into the brain, then a hypertonic solution is created, and then water is like pumped into this. Okay, and then isotonic. In an isotonic solution, the concentration of water on the inside and the outside is exactly the same, so there is no net movement of water, and that's a situation called homeostasis. Okay, so you'll have to explain it in all of these 3 sets in terms of all these terms, like osmotic potential, water potential, and then hypotonic, hypertonic, and isotonic. And I think you'll be able to do that by looking again and again at the video. Thank you.